My name is Jarad Mason, and I'm an assistant professor in the Department of Chemistry and Chemical Biology at Harvard University. I received a Beckman Young Investigator Award from the Arnold and Mabel Beckman Foundation in 2019. Dr. Arnold O. Beckman was a chemist, an inventor, and a philanthropist. He made countless contributions to advancing scientific research and helped enable many of the technologies that underpin our modern society. In particular, the instruments that Dr. Beckman developed over the course of his career, which included pH probes, spectrometers, and potentiostats, have allowed scientists and engineers to investigate how the structure and composition of different molecules and materials impact their properties. This has accelerated the development of a whole host of important chemical compounds, including new pharmaceuticals and advanced materials for catalysis and clean energy. Certainly, much of the research in my group at Harvard would not be possible without many of the instruments and tools that were initially developed and commercialized by Dr. Beckman. Funding from the Beckman Young Investigator Award enabled my laboratory to pursue a completely new research direction. Several years ago, my students and I became interested in trying to find ways of tackling a very old but important challenge related to transporting gases in water. Water is a critical solvent for many industrial reactions, as well as for all biological processes. But water is a very polar, strongly associated solvent that does not like to make space for less polar substances, like many small gas molecules. As a result, the solubility of most gases in water is extremely low, and this is a big problem for many energy and biomedical technologies that require gas molecules to be transported within an aqueous environment. As an example, if you imagine a glass of water sitting on your counter, there will be just a single oxygen molecule in every 8,000 cubic nanometers of space. This density is roughly equivalent to an office building with just a single person in every 5,000 cubicles, and is not a high enough concentration of oxygen to keep multicellular organisms, like us, alive. This is why we evolved over billions of years hemoglobin, red blood cells, and a circulatory system so that we could transport high enough densities of oxygen and water to sustain human life. Though biology solved the oxygen transport challenge, at least when everything is working properly, the low solubility of oxygen and other small gas molecules in water still remains a major challenge for many technologies. This includes, for example, electrocatalytic reactions that are currently being developed to reduce carbon dioxide into liquid fuels and to reduce nitrogen into ammonia. In these reactions, carbon dioxide and nitrogen have to be transported from the gas phase through an aqueous electrolyte to the surfaces of an electrode where they react. Low carbon dioxide and nitrogen solubilities in the electrolyte lead to severe mass transport limitations that reduce how quickly products can be generated during these reactions. Leveraging funding from the Arnold and Mabel Beckman Foundation, my research group recently developed a new approach to transporting gas molecules at high densities in water. Our approach relies on using different classes of microporous materials, which are materials that contain tiny, roughly 1 nanometer pores that can lead to extremely high internal surface areas. More surface area than a football field in just a half teaspoon or so of material. These surfaces have attractive interactions with gas molecules that allow gases to be concentrated through surface absorption to much higher densities than can be dissolved within a liquid, or even than exist in the bulk gas phase. Normally, however, when you take a porous material and put it in water, water fills the pores and prevents gas molecules from absorbing. So most solutions of microporous materials actually contain fewer gas molecules than pure water. My group had the relatively simple idea that we could get around this by designing microporous particles with very hydrophobic internal surfaces that make it thermodynamically unfavorable for liquid water to enter, but also very hydrophilic external surfaces that allow the particles to form uniform, stable dispersions in a variety of aqueous solvents. Using this approach and building on a lot of important previous research in porous materials, nanoscience, and biology, we were able to make aqueous solutions that look like water but contain high concentrations of microporous particles with dry pores. We call these solutions microporous water. Even though the particles in the solutions are surrounded by water, liquid water does not go inside the pores, but gas molecules do. This leads to huge enhancements in the density of gases within the solution. For example, we have now been able to make aqueous solutions that have less than 4 volume% of microporous particles, but carry the same amount of oxygen as blood. This opens up exciting opportunities for many biomedical applications, including the development of artificial blood substitutes, injectable sources of oxygen, and media for organ preservation that my group is now actively pursuing. I'm very appreciative of the support my group received from the Beckman Young Investigator Award, which enabled us to start down this new research direction.